Well, good day to everyone joining us and welcome to today's XTalks webinar. Today's talk is entitled Adaptive Immune Response in Narcolepsy Type 1. My name is Sumaya Sheikh and I'll be your XTalks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, you can contact me at any time by sending a message using this chat panel. We also have 2 handouts available for this presentation, so audience members, you can download them through the handouts tab in your control panel. At this time, all participants are in listen-only mode. Please also note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank GenScript who developed the content for this presentation. GenScript is the leading contract research organization in the world, providing gene, peptide, protein, CRISPR, and antibody manufacturing services. Since its foundation in 2002, GenScript has grown exponentially through partnerships with scientists conducting fundamental life science research, translational biomedical research, and early-stage pharmaceutical development. The company is recognized as having built a best-in-class capacity and capability for biological research services encompassing gene synthesis, peptide synthesis, custom antibody and protein engineering, and in vitro and in vivo pharmacology, all with the goal to make research easy. Now it's my pleasure to introduce our speaker for today's event, and that's Dr. Gu Liu. Dr. Gu Liu is an instructor at Stanford University School of Medicine and an immunologist with a special interest in T-cell immunology and brain disorders. His projects involve isolating and characterizing T-cells reactive to autoantigens and flu antigens in neurological disorders using state-of-the-art technologies including DNA barcoded tetramers, dextramers, next-generation single-cell sequencing, and modeling of T-cell receptors, so that's TCRs. Most of his past work is applied to narcolepsy, an autoimmune disease associated with human leukocyte antigen, that's HLA-DQ0602, that is triggered by the flu and involves flu hypocretin orexin cross-reactive CD4 plus T cells. So now, without further ado, I'd like to pass control to our speaker. So Dr. Liu, whenever you're ready, you can go ahead and get started.
Yes. Good morning, everyone. And thanks, GenScript, for this opportunity to present my work. It's about the adaptive immunity in the narcolepsy type 1 and the neurodegenerative disorders, Alzheimer's and Parkinson's diseases. So narcolepsy is a lifelong neurological sleep disorder. It affects the brain's ability to control sleep-wake cycles. The patients with narcolepsy fall asleep frequently and involuntarily during their daily activities. The narcolepsy affects 25 to 50 out of 100,000 people in the world, and there are 2 types of the narcolepsy, type 1 and type 2. And the patients with narcolepsy type 1 or NT1 have low levels of the hypocretins in their brains or experience cataplexy, which is a temporary muscle weakness or loss of muscular controls. And the cataplexy is triggered by strong but usually positive emotions such as joking and laughing. And there's no cure for narcolepsy currently, but some symptoms can be treated by medicines and lifestyle change. So narcolepsy type 1 or NT1 has been well characterized with the loss of the small group of the neurons in the brain. So these neurons are highly expressed hypocreatins, so also known as hypocretin neurons. Hypocretins are small neuropeptides that regulate wakefulness, arousal, and appetite. There are two types of the hypocretins, hypocretin 1 and hypocretin 2. So they are cleaved from the same precursor and shared almost identical C-terminal antigens. There's no specific cause for narcolepsy type 2, so I focus on narcolepsy type 1 in this research. So after many years of the research, the mechanisms of the hypocretin neuron loss in the narcolepsy type 1 is still not clear. One trigger of the narcolepsy type 1 is that the influenza A H1N1 infection and a pandemic vaccine in 2009 to 2010. Their segment HA and NA are located on the viral memory, and they are the primary antigens that are recognized by immune system and are capable of triggering immune response, including the antibody seeker production that can fight an infection. Inside of the virome, the AMPs play a central role in viral replication, and it's the most abundant viral protein in the infected cells, and thus is an ideal target to develop the next generation antiviral drug. The genome-wide association studies have identified several genetic risk factors. The human leukocyte antigen HLA, DQA1, O102, DQB1, O602, or DQ0602, and T-cell receptors, or TCR, alpha chain TRA are the two most significant risk factors. In adaptive immune system, the HLA is very important. It presents antigens to TCR, initiating the immune response. There are two types of the HLA, class 1 and class 2. The class 1 HLA presents antigen to cytotoxic CD8 T cells, while class 2 HLA presents antigen to TCRs of the CD4 helper T cells. So each HLA is a heterodimer, and almost every nucleated cell expresses class 1, while class 2 are usually present only on the professional antigen-presenting cells, like B cells, monocytes, macrophages, dendritic cells, and activated lymphocytes. Class 1 HLA consists of a happy chain which is mainly encoded by ABC genes and extracellular beta-2 microglobulin. Its binding group accommodate a peptide with 8 to 10 but predominantly 9 amino acids. Class 2 HLA consists of alpha and beta chains, which are mainly encoded by DR, GP, and DQ genes. They have the overall same structure, and its binding group. Class 2 HLA has a open group that can accommodate a longer peptide with 12 or more amino acids. The amino acid variations in several regions can change the fine shape of the groove and thus alter the peptide binding specificity. Many TCR, over 40,000 HLA alleles have been reported, each with of their own nomenclature. Certain diseases, especially of autoimmune disorders, are associated with particular HLA types. Among the most prominent associations are the AS with the class 1 B27 and celiac disease with the class 2 DQB102 and narcolepsy with the class 2 DQB10602. TCRs is super polymorphic too. and an individual has millions of billions of TCR clones at an average. And there are two forms of the TCRs. The alpha beta TCRs is present on more than 85% of the peripheral blood T cells. The generation of the TCR diversity is similar to that for antibodies. Each TCR is mainly produced by genetic recombination of VDG genes. So each TCR is composed of two different alpha beta chains, and each chain is composed of two extracellular domains, the variable region and the constant region. The variable domain of each TCR has three complementary determining regions or CDRs. The CDR3 directly contacts with the presenting antigen and is mainly responsible for recognizing it. And therefore, it has the most sequence polymorphisms. The TCR binding to class 1 HLA with a stronger affinity compared to class 2, which could be due to the structural difference in peptide presentation between class 1 and class 2. The adaptive immune response is antigen-specific. The full activation of the adaptive immune response requires both signal 1 and signal 2. The antigen presentation by HLA to TCR serves as signal 1. With signal 2, the CD28 on B cells binding to B7 and triggering different downstream pathways like F8 infed, F-kappa B, and AP1 pathways, inducing the secretion of the cytokines and chemokines, expansion of the helper CD4 T cells. This immune response could be inhibited by multiple pathways, one of which is the CTLA-4 on T cells binding to B7 and blocking the signal 2. The binding affinity of CTLA-4 to B7 is much higher than that of the CD28. Other tolerances include the energy and TCR just bind to the HLA and FOXP3 expressing Tregs and apoptosis. The adaptive immune system protects us from infection, and given the complexity of this system and all different kinds of self and foreign antigens and the polymorphisms of the TCR and the HLA, many programs have been developed to predict the HLA peptide binding and TCR clustering, especially using the deep learning. As the narcolepsy type 1 is highly associated with the HLA and the TCR, we hypothesize there's an adaptive immune response to hypocretin and pandemerics, and molecular mimicry between them. So first, to find the potential antigens, I screen the overlapping peptide from the key self and foreign antigens using a competition binding method. Each peptide has 15 amino acids in length. In this binding method, a biotin-related peptide, which is known to bind to DKO602, is co-incubated with the unbiotin-related peptide and DKO602. The reaction was transferred to a 96-well plate pre-coated with anti-DKO602 antibodies, and the EU labeled streptin evidence was added to each well, and a time result of fluorescence is developed. The biotin-related peptide alone is a positive control, and other competitor peptide is compared to it. So therefore, the lower fluorescence readings indicates the stronger binding. I set the 25% of the biotin-related peptide binding as the threshold of the strong bindings. The autoantigens include the hypocretin and the RFS4, which is a transcription factor co-located with the hypocretin. Foreign antigens include the pandemerics segment HA, NA, PB1, and MP. Importantly, some peptides with the post-translational modifications or PTMs are also tested, including the C-terminal amidation and the phosphorylation. So overall, a total of over 2,000 peptides that are synthesized by GenScript are tested, and around 300 strong binders were detected. So to further investigate these strong binders, I studied the cell culture of 6 NT1 patients and 4 controls using strong binders only from hypocretin, HLA, and AMP, RFS4 as a control. To increase the sensitivity of the TCR binding to the HLA, I generated the tetramer DKO602 loaded with each of these strong binders. So meanwhile, the peripheral blood mononuclear cells, or PBMCs, were stimulated with the cognate peptide and stained with the tetramer DKO602 and stained with the T-cell surface markers CD3, CD4, and CD8 labeled with the distinct fluorescence. The cell cultures were harvested and analyzed with the flow cytometry. So ideally, a staining pattern with the clearly separated populations of the tetramer positive CD4 T cells indicates a positive immune reaction to a peptide in an individual. These cells are antigen-restricted CD4 T cells. So after looking at all these strong binders, all these staining patterns, we can see that the more individuals have positive immune reactions to foreign antigens, than to autoantigens. And the frequency of the antigen-restricted CD4 T cells indicates a strong or weak immune response. To investigate the immune difference between patients and controls, the 7 peptides were selected because most of these 10 subjects have positive immune reactions to them, or they have the sequence similarity. With a few more subjects, I found there's no difference for 3 of them. And for the rest 4 TCR peptides, 2 from the Pandemrix HA629 and MP136, 2 from the C-terminal hypocretins, hypocretin 128 and 226. Only when modified with the C-terminal amidation, I tested over 70 NT1 patients and 40 controls. Using tetramer DKO602 loaded with each of these 4 peptides, the PBMCs were stimulated with the whole Pandemrix vaccine and a peptide in parallel. So the frequency of the antigen-restricted CD4 T cells is significantly higher in narcolepsy type 1 patients than that in controls with the HA629, MP136, and hypocretin 128 when stimulated with the Pandemrix vaccine and a peptide, but not for the hypocretin 226. It suggests there's an autoimmune response to hypocretin. These antigen-restricted CD4 T cells were also detected with the dextramer, which is more sensitive. These cells were sorted out for single-cell sequence for both the TCR and the whole transcriptome. So over 20,000 TCR clones were recovered, and the TCR clone that detected in more than one cell means enrichment. The enrichment for some TCR clones are up to hundreds of times, and they are detected across peptide and subjects between patients and controls. It's higher for Pandemrix peptide compared to hypocretin, and the enrichment in the same subject suggests the T cells are clonally expanded to a peptide. And a mimicry hypothesis is that there's some TCR clones that could be cross-reactive to the Pandemrix peptide hypocretin. So to test it, TCRs are transfected into a human-injected cell lines that doesn't express any other TCR. This cell lines also engineered with luciferase expressed under infect pathway. So these cell lines then co-cultured with a peptide and artificial antigen-presenting cells that express DKO602 only. The photo change of the luminescence is compared between the peptide and the background a threshold of the 1.2 is set to be positive. And a total of the 709 TCRs were transfected and tested. Some of them are highly enriched, and some of them are chosen with the priority in hypocretin isolation. And I found 103, 89, and 37 TCR clones are activated by HLA-69, MP136, and hypocretin, respectively. And their sequence similarity of the CDR3 sequence shows that the TCRs activated by HLA-69 or MP136 is clearly grouped into the major clades or clusters, and TCRs activated by hypocretin fall into them as a single clone. So here red is activated by HLA-69 and green by MP136, and hypocretin is blue. These activated TCR clones include many with TRAG24, 28, and TRBV4.2, which are associated with narcolepsy type 1 as found in GWAS. And 95% of the activated TCR clones are detected at least once. So also another 100 TCR clones are found over 10 times. They were not they couldn't be activated by any of these antigens. So there are multiple possibilities. One of is that it's they just bind the TCR like energy. So as shown here, we can see that in one example, this actually restricted CD4 T cells is clearly separated, and 70% of these cells are just CD69 negative, which is an early activation marker. So furthermore, I selected 28 TCRs, include a few negative, for testing activation with more peptides. And interestingly, I found 5 of them are activated by HLA-69 and another HLA peptide, one by MP136 and another MP peptide, and 7 by hypocretin peptide with different lengths of the amino acid, and including the full length of the hypocretin 1 and 2, and a peptide containing the binding core without C-terminal amidation. In spite of the sequence similarity, no TCR clones was found to be cross-reactive to both pandemerics and hypocretin. So for the single cells isolated for tetramer, they were sorted into 96 well played and sequenced for about 20 cell markers of interest. And for each activated TCR clones, the average of the gene expression among the cells have the same TCR clones in one subject were calculated and compared between patients and controls. For negative TCRs, the average was made with all the cells into diagnosis groups. The average of the activated TCR clones for each peptide is further calculated. With the dextramer, cells were sorted in bulk and sequenced with for the whole transcriptome with the 10x pipeline. And only cells with paired TCR alpha and beta chains were clusters with UMAP, and the tested TCR clones injected cell line were relocated. So it suggests a role of the cytotoxic CD4 T cells with both tetramer and dextramer. To further investigate the pathophysiology of the NQ1, I explored 3 different directions. So first, the hypocretin neurons express other neuropeptide and proteins. So I tested the immune response to RFS4 and found a few subjects that could react to some of these peptides, but there's no difference between patients and controls, and there's no cross-reaction TCR clones between the RFS4, pandemerics, and hypocretins. So second, the PBMCs at baseline without culture is close to its in vivo physiology to investigate the antigen-restricted CD4 T cells at baseline. I pulled multiple PBMCs from multiple NT1 patients and controls, and enriched the CD4 T cells using a magnetic base. So this enriched CD4 T cells were next incubated with the tetramer, with the tetramer GQ602 loaded with the HA69 MP136 and hypocreatine. The antigen-restricted CD4 T cells were detected and sorted for single-cell sequencing. And a total of 430 TCR clones were recovered, and 5% of them were detected in more than one cell and meaning enrichment. And 56 TCR clones were detected at both baseline and cultured PBMCs, and 40% of them was activated by a pandemics peptide in the previous test injected cell line. So these cells were further clustered using a UMAP, and the tested TCR clones were relocated. Most of these cells have paired alpha and beta chains, and the further analysis for the phenotyping is ongoing. So second, and lastly, and the cerebrospinal fluid sample or CSF samples are very important to find a difference between patients and controls. So I performed the single-cell sequencing with some, and I recovered around 500 cells from 6 patients and 6 controls using a 10x technique, and I recovered around 6,000 cells from 16 patients and 15 controls using a BD technique. So most of these cells are lymphocyte, and the populations of the CD4, CD8 monocytes and B cells were determined. There's a slight enrichment for a few TCR clones, and the gene suppression suggests a role of the cytotoxic lymphocyte as one of the cell markers like GNLY is differentially expressed between patients and controls. So taking away from the narcolepsy type 1 that we found many strong binders from both pandemics and the autoantigens, and there's differential immune response to hypocreatine and C-terminal hypocreatine only when modified with the amidation, and to pandemics peptide HA69 and MP136. Sorry, there's a type. And many TCRs are activated by one of these peptide, and including the GWAS-associated TRA24 and TRBV4.2, and no cross-reactive TCRs so far. And there's a role of the cytotoxic CD4 T cells, T lymphocyte in the narcolepsy type 1. So I also did lateral research about the neurodegenerative disorders. The Alzheimer's disease, or AD, has long been characterized with 2 hallmarks in the brain: the neurofibrillary tangles in the neurons and extracellular amyloid plaque, which are composed of the hyperphosphorylated tau and the amyloid beta. The neurodegenerative disorders, of which the AD is the most common, that are associated with the accumulation and aggregation of the abnormal misfolded tau in the brain, are collectively termed as tauopathy, and suggesting that tau playing a central pathological role in these disorders. The major role of the tau is promoting the microtubule assembly and stability. Normally, tau is soluble and natively unfolded, so whereas it becomes insoluble when hyperphosphorylated and detached from microtube and self-aggregated into the paired helical filament, or PHF, oligomers and tangles. So there are many other PTMs at different sites that may influence the function and pathology of the tau, including the acetylation and methylation and ubiquitination. So it has become clear in the past 2 decades that adaptive immune system, of which T cells are a major part, does act in the brain. So multiple groups describe the role of the T cells in the brain of the neurodegenerative disorders and AD. So the GWAS has identified many genes with potential roles in adaptive immunity, and including the HLA. So one question is that which HLA specific allele is playing. So our large cohort and multi-ancestry GWAS identified a shared SMP signal between the AD and Parkinson's disease or PD, and a hierarchical protective effect of the DRB104 alleles. It's the strongest with the DRB104, and 0407, intermediate with the 0401 and 0403, absent for DRB10405. The same signal is also associated with the decreased neurofibrillary tangles in postmortem brains, and the more strongly associated with the tau levels in CSF samples. So we hypothesize there's an adaptive immune response to this tau. So first, I tested the binding affinity of the overlapping tau peptide, and including the most frequently PTMs, including the phosphorylation, acetylation, and methylation. So in priority, I tested with binding affinity with against the DRB10404, 0401, and the DRB10405 at a high concentration of the peptide. So we found there are 3 regions that bind to DRB10401, 0404, but not DRB10405. So they are the N terminals, and C terminals with and without phosphorylation, and PHF6 only when the K311 is acetylated. So the dose response was further tested again, including 2 more alleles, DRB10403 and 0406, and confirmed their binding affinity. The PHF6 is playing a dominant role in tau self-aggregation, and I tested a few AD samples with this tau PHF6 AK311, and using tetramer DRB10401 and 0404 as to their HLA phenotypes, and in parallel with some PD samples and cognitive health controls. The frequency showed no difference in this small sample size, and but it's with a higher frequency in DRB104 subjects than that DRB104 0401. So these cells were single cell were sorted into a 96 well plate, and 4 plates were sequenced with the SMART-seq technique, which is with the ultimate sensitivity. And these TCRs were clustered after filter out this monocyte and some background. So multiple TCR clones were found at least 1 cell, especially for these AD patients and these healthy controls, and 90% of them have the same TCR clones, and the gene suppression showed here the FOXP3 is highly expressed in AD patients, is suggesting a role of the Tregs. So further investigate if this TCR clones is activated by tau PHF6 peptide. I cloned these TCRs and HLAs into a vector, upstream the GFP, and generated the lentivirus in 293 T cells, and transfected them to different cell lines. One is the artificial antigen presenting cell line, and the jerked 76 cell line that doesn't express any other TCR, and featured luciferase expression and infect pathway. These cells were purified with the flow cytometry and co-cultured with the tau peptide, and the luciferase activity will be developed. So this is our proposed the clearance of the role of the DRB104 in the in AD. So it's present the tau PHF6 when isolated at H3K311 is presented by DRB104 to the T cells and then mediate the immune response. With the Parkinson or PD samples, I did the similar experiment, but with the alpha-synuclein. I found there's one region in the C-terminal that bind to DRB10404 and 0401, but not a 0405, which is consistent with the GWAS association. So, and the C-terminal, the binding affinity is slightly decreased when phosphorylated tyrosine 125. And with a small sample size, I stimulated a few the PBMCs using a pool of these 4 peptides, and they are labeled the same fluorescence. And there's a higher frequency in DRB104 PD when, yes. And to further investigate this each single peptide, I use labeled each tetramer, DR tetramer with unique fluorescence, and the PBMCs were stimulated with still the pole of this peptide, but stained with this tetramers, so that the specificity of each single peptide can be investigated. So I have stained with many hundreds of the stainings with different from DRB104 and 04. DRB104 positive subjects with the different pole peptide from tau and alpha-synuclein, and the analysis is ongoing. So a summary about this AD and PD, we found the hierarchical protective effect of the DRB104, which may involve in the protective and the clearance of the tau in AD, and the PHF6 less than 311 only when acetylated could bind to DRB104, and 0401 but not DRB10405. And there's a C-terminal region of the alpha-synuclein does that too. And we also detected this AK311 alpha-synuclein antigen-specific CD4 T cells in them. And detective recovered some TCR clones that are clonally expanded. And so we have some projects is ongoing experiment, like we investigate other self-antigens, neuropeptide for the and different other influenza B and CSF samples in narcolepsy type 1, and investigate the antigen-specific CD4 T cells in baseline PBMCs, and test the immune difference with more AD and PD samples, and also make transfection in mouse models to see how it works. And I would like to thank our collaborators and the with Dr. Minot lab and Garicia's lab and GenOR lab. They provide us like many samples and reagents. And thanks for your attention.
Well, thank you very much, Dr. Gu, for that fantastic presentation. Now it does bring us to your Q&A. So audience members, if you do have more questions, you can keep sending them in. We've already received some questions, so let's start off with those. All right, so our first question, Dr. Liu, is, have you tested out more binders for narcolepsy type 1?
Yes, that's a good question. As shown in the slide, we have found 300 strong binders, but I mean, there are many others to be potentially having the difference, immune difference between patients and controls. And one peptide from the pandemic segment NA, like NA55, and has shown that there are some potential in immune difference. As the antibody level in the influenza B, we already knowing that, but not published. There's the difference in the immune, the antibody levels. So, and then the NA55, that peptide is shared between the influenza A and B. That's what we are working on, and also we know the strong binders is the threshold of 25. There are many others, like between 25 and 50%. There are still a lot of work to do for many the binders. Yep.
Great. Thank you, Dr. Liu, for that answer. And we have another interesting question from an audience member. That question is, why do you think there's no cross reaction between foreign and AUO antigens?
Oh, that's a great question. Yes. So the cross reaction, the first the hypothesis we did is that some TCRs could be react both the pandemerics, the peptide, and hypocretin. So we did the transfection and tested over 700, but none of them could be cross-reactive. There are multiple possibilities, and once that is the T cell energy, like the TCR, they just bind to the HLA without further downstream signaling. So that's why you can see there are many other when I did the tetramer staining, it's only the technical signal one, the tetramer GQ62 present antigen to HLA. And the secondly, it could be that these cells is Tregs, the highly expressed FOXP3 and the CTLA4 that they inhibited the immune response. And another one is that it could be these cells are involve the apoptosis in the further. So yeah, but there are still many other pathways, like because the TCR, the Jekyll TCR features in fat pathway, but there are many other pathways in T cells that we haven't tested. Yes, that's it. Thank you.
Thank you. Thank you for that comprehensive answer. And we have another fantastic question from an audience member. So that question is, have you tried other two binders of TAU?
Yes, there are 3 regions that bind to the DRB1 0404 and 0401. This is N-terminal and C-terminal. The C-terminal with and without phosphorylations. So in the further testing as tetramer staining, I labeled them both the C-terminal and N-terminal, and together in parallel with the PHF6, labeled all these 4 peptides with unique fluorescence, so that when you stain the samples with the pole of them, and then when do the flow cytometry analysis, you can see for each channel corresponding to each peptide, we can investigate the difference, the frequency, and the single cells. So they are on the. So we are doing that right now, currently, yes.
Fantastic. Well, thank you, Dr. Liu, for all of those answers and for all the questions that came in from the audience members. Thank you so much for sending those over. If you do have more questions, not to worry, you can reach out to the team at GenScript. So here is the contact information. Go ahead, quickly jot that down. So that's peptide@genscript.com. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us improve our webinars. Additionally, I just sent a link of the recording, so where the recording will be uploaded. I've sent that in the chat box, so go ahead and also take that down, and it generally takes us 1 to 3 business days after the webinar for that recording to be uploaded. Now, please join us in giving a big thank you to our speaker today, Dr. Gu Lu, for that fantastic presentation. We hope you found this webinar informative. Have a great day, everyone.
Thank you.